Hey you guys, here we are talking about the electron transport chain, or 9.4. Um, so let's first of all get a big picture. This is our mitochondria. So this is going to be the outer membrane, which fades out, and then this is going to be our inner membrane right here. So on the inside, this inner mitochondrial matrix, this is where the citric acid cycle is happening. This is where pyruvate processing is happening. And so here we are making the NADHs and we're shipping them over to the electron transport chain, or over to the inner membrane, because this is where the electron transport chain happens. And so the big idea is this, that electrons pass through the inner membrane. They never go into the intermembrane space. So the electrons pass through the inner membrane and come back into the matrix, whereas hydrogen ions get pumped into the intermembrane space. So if you think about it, hydrogen ions are the basis of acids, and so this space is a little bit more acidic than other parts of the mitochondria, because we're constantly pumping hydrogen ions into this space. So that's the big idea. Now, the electron transport chain is composed of 4 proteins, or protein complexes. So a protein complex is going to have coenzymes or cofactors, and it's going to be multiple proteins put together. And so we have 4 of these protein complexes, 1, 2, 3, 4. And at the very end, we have a half of an oxygen molecule, or 1 oxygen atom, so 1/2 O2. And what's pulling these electrons through the electron transport chain is that this 4th protein complex is very electronegative. The 3rd is a little bit less electronegative, and the 2nd is a little bit less, and so forth. And so this electron gets donated right here at the beginning. The NADH is going to donate electrons, and the electron is going to pass through these proteins in the inner membrane, and it's going to be pulled by the increasing electronegativity. Remember that electronegative is an affinity, or a pull on electrons. And then at the very end of this 4th protein that is very electronegative, there's a half of an oxygen molecule, or one oxygen atom, which is extremely electronegative. And so it's driving this electron to pass through all of these proteins. And so this is a spontaneous reaction because it is electronegativity that is pulling all of these electrons through. And so at the very end of this, that electron attaches to the oxygen, and then that oxygen is going to attach to hydrogens, or hydrogen ions that exist out here, and they're going to create water, and then we breathe that water out. It's our low energy end product. But let's follow that through later. So here at the first protein, NADH is going to be donated. And so NADH is going to be donated right here where we see the FMN, and it's going to pass through all 4 of these proteins. FADH is going to be interacting with the second protein. And so FADH, because it is working with the second protein complex, it is going to allow fewer hydrogens through. You will understand this later, but it is a lesser tool because it can only because it's halfway down the chain. This will make more sense in a moment. And so here, the electron is going to be donated at this FMN, or this prosthetic group, the flavin mononucleotide right here. So that flavin mononucleotide is going to pass it down to a sulfur iron containing prosthetic group. And so this electron is going to pass through, so it's going to, this protein complex is going to be reduced for a moment because it's going to gain. Remember, reduce is gain. It's going to gain an electron. And then it's going to go back and become oxidized because it's losing an electron as the electron passes into the second protein complex. This second protein complex is going to be reduced for a second, and then it's going to pass the electron on and it's going to be oxidized again. This third protein is going to be reduced for a second, and then it's going to become oxidized as it passes on the electron to the fourth, and so on. And so here, when the electron is reduced, it allows a hydrogen ion to pass through. And so the FADH is going to allow for, so when an electron is donated from FADH to the second protein complex, it's going to it's going to pass through or reduce only 3 proteins, whereas NADH is going to pass through 4 proteins and then attach to the oxygen and attach to hydrogen to make water. And so here, the second protein complex has a part of it called ubiquinone or cytochrome or coenzyme Q, and then it's going to pass on to the cytochromes. The protein complex 3 and 4 are filled with just cytochromes. When cytochromes are proteins that have a heme group or a prosthetic group, like your hemoglobin. And so here, remember that our last electron acceptor is oxygen. And so I think that you have the basic idea of electrons now. After the electron binds to the oxygen molecule, here, so let's follow this, the NADH is going to donate the electron. It's going to pass through the 4 proteins, and then here the electron is going to bind to this oxygen molecule back in the mitochondrial matrix, and then it's going to attach to the hydrogen ion here in the mitochondrial matrix, and then this water is going to pass through, and it is going to be it's going to pass through our blood, and we're going to breathe it out. And so this is why we breathe out water right here. So here, let's look. Remember that NADH and FADH donate both electrons and protons to the electron transport chain. So notice that the electrons stay within the membrane, whereas the hydrogen ions are going to be pumped through into the intermembrane space. So here, there's our 1, 2, 3, 4 proteins, just a different view. And so what's going to happen is that we're going to use these electrons to reduce these proteins complexes. And when the protein complexes are reduced, it allows the hydrogen ions that are being donated by NADH to pump into this intermembrane space. You're going to have a whole bunch of hydrogen ions in here, and when you have a whole bunch of hydrogen ions here, it's going to create a proton gradient or proton motive force, which is just simply a concentration gradient. You have a whole bunch of hydrogen ions, and if you were to have a hole in this membrane, you would have then a concentration gradient, and the hydrogens would move through, would pass through from high to low concentration. But instead of a hole in the membrane, what we have is an enzyme, an ATP synthase, which allows the hydrogen ions to pass through. And so here's our hydrogen ion, which will eventually become water or used for something else. And this hydrogen ion is going to pass through the ATP synthase, and it's going to spin it. So it's just sort of like a crank. Every time the hydrogen ion goes through ATP synthase, it spins it a little bit. And scientists are not totally sure, this is debated, but they believe about 4 hydrogen ions are going to make a complete circle. And when we have a complete circle, ADP donates a phosphate group to ATP to make ATP. And so this is why we call it ATP synthase, and this is oxidative phosphorylation. We call it oxidative phosphorylation because remember, this whole thing is driven by this oxygen that's pulling the electrons through the protein complexes because it is so electronegative. It is this electron that's being passed through that's reducing the protein complexes to allow hydrogen to pass through. And so here we call it oxidative phosphorylation because it is being driven by an oxygen. And so it is about 2.5 NADH can create about 1 ATP because it's about 4 hydrogen ions to go through the ATP synthase. But that's not necessary to remember. And so let's come back to the concept of energetic coupling. The proton motive force, or chemiosmosis, is an exergonic process, and it is what drives the endergonic process of ATP synthesis. And so chemiosmosis, this is the first time that we're talking about it, but it is also found in other organelles or other processes such as photosynthesis, which we will talk about later. I hope this helps. Please let me know if you have any questions.